A quiet revolution is taking place in the world of medicine. Far from the dramatic surgical theaters or bustling emergency rooms, this movement unfolds in pristine labs, in the invisible dance of molecules, and within the DNA strands curled tightly in every cell. It is a revolution not aimed at curing disease in the traditional sense, but at bending the rules of life itself, specifically the rules of aging. For centuries, aging has been accepted as an inevitable process. Wrinkles deepen, joints stiffen, memory fades, and eventually, the body surrenders. But a new wave of science argues otherwise. In this new paradigm, aging is not a passive consequence of time, but an active process, one that can be studied, manipulated, and perhaps even reversed. One of the most promising fields in this longevity frontier involves senolytics. These are a class of compounds designed to selectively destroy senescent cells, the so-called zombie cells, that stop dividing but refuse to die. These cells accumulate with age and secrete a toxic cocktail of inflammatory signals, growth factors, and enzymes, collectively referred to as the senescence-associated secretory phenotype, or SASE. This SASP is thought to drive many age-related diseases, from arthritis to atherosclerosis to cancer. But beyond disease, it also appears to accelerate the underlying process of aging itself. What if we could clear out these zombie cells, like pruning dead branches from a tree? That's exactly what senolytics aim to do. In mouse studies, senolytic treatments have produced remarkable results. Old mice treated with a combination of dasatinib and quercetin, two of the most studied senolytics, exhibited increased physical function, better cardiovascular health, and even longer lifespans. Their fur grew shinier, their kidneys functioned better, and they moved with a youthful energy. But this is not just about looking younger. It's about improving health span, the period of life spent in good health. Human trials are already underway. A small 2019 pilot study showed that a senolytic cocktail improved physical function in patients with idiopathic pulmonary fibrosis, a devastating age-related lung condition. But perhaps even more compelling is the possibility that senolytics could reduce epigenetic age, a biological marker that many scientists believe to be a more accurate indicator of aging than the number of candles on your birthday cake. Epigenetic age refers to the patterns of chemical markers that attach to DNA and regulate how genes are turned on or off. As we age, these patterns change in a predictable way. Scientists have developed so-called epigenetic clocks that can estimate a person's biological age based on these changes. In recent studies, some senolytic treatments have been shown to reduce the epigenetic age of blood samples. It's as if the molecular signature of the cells reverts to a more youthful state. This is nothing short of revolutionary. For the first time in history, we may be able to quantify aging and watch it slow or even reverse them in real time. But senolytics are only part of the picture. If removing old cells is one pillar of rejuvenation, then restoring youthful function is the other. And that's where cellular reprogramming enters the story. Inspired by the discovery of Yamanaka factors, a set of 4 genes that can turn adult cells back into stem cells, researchers are exploring how partial reprogramming might erase the hallmarks of aging without erasing cell identity. In 2020, scientists at the Salk Institute used these factors to restore vision in old mice with damaged retinas. The technique didn't just improve sight. It actually rejuvenated the cells at the molecular level. Their epigenetic age rolled back. Their mitochondria began working more efficiently, and inflammatory signals dropped. This is still early-stage science, and the road to safe, controlled reprogramming in humans is long. But the idea is seductive. If we can hit refresh on the body's aging cells, perhaps we can treat not just disease, but aging itself as a modifiable condition. Still, there is another realm of the body being explored with equal excitement, a domain so fundamental that it has shaped our evolution from the very beginning, yet so foreign that most people are unaware it even exists inside them. That realm is the microbiome. Trillions of bacteria, viruses, fungi, and other microbes reside in and on our bodies, with the gut hosting the densest population. Far from being mere passengers, these microorganisms are now understood to be integral to human health, influencing everything from digestion and metabolism to immunity and even mood. What's increasingly clear is that the microbiome also plays a critical role in aging. As people age, their gut microbiota tends to lose diversity. The balance between beneficial and harmful bacteria shifts. Inflammaging, a chronic, low-grade inflammation common in older adults, has been linked to this microbial imbalance. But can changing the microbiome actually affect the aging process itself? A series of studies in recent years suggest it might. In a groundbreaking experiment, scientists transplanted fecal microbiota from young mice into old ones. The results were astonishing. Not only did the older mice show improvements in cognitive function and memory, but their brains began to look younger under the microscope. Synaptic plasticity improved. Markers of inflammation decreased. The old mice, in effect, became mentally sharper and biologically younger. But mice are not humans. So, researchers turned to humans. In 2022, a pilot study explored fecal microbiota transplantation, FMT, in elderly patients with mild cognitive impairment. Though preliminary, early results indicated improved gut diversity and reductions in markers of systemic inflammation. Researchers are now working to refine the protocols. What donor traits produce the most benefit? How often should transplants be performed? Are oral capsules as effective as enemas? These questions remain, but one thing is clear. The microbiome is no longer a footnote in the story of human aging. It is a central character. The implications are profound. If the gut can influence the brain, and if both can be rejuvenated through targeted interventions, then aging may be far more plastic than previously imagined. Imagine a future where people undergo regular microbiome tuning, like changing the oil in a car, or where senolytic therapies are administered prophylactically in middle age to forestall the onset of disease. Imagine a doctor telling you not just that your blood pressure looks good, but that your biological age has decreased by 3 years since your last visit. This is not science fiction. This is the trajectory of current science. Critics are right to urge caution. The body is an exquisitely complex system, and tampering with its balance could have unintended consequences. Senolytics might clear beneficial senescent cells that help in wound healing. Microbiome transplants might carry unknown viruses or pathogens. Reprogramming cells could cause cancer if misapplied. But every medical revolution has begun with bold, sometimes risky steps. Penicillin once seemed like alchemy. Organ transplants were once considered madness. Today, they are standard practice. As anti-aging research matures, it will need to grapple with ethical and philosophical questions as well. What does it mean to live longer lives, not just for individuals, but for society? How do we ensure these therapies are accessible to all, and not just the wealthy few? Will increasing life expectancy exacerbate overpopulation, or will it create a new golden era of wisdom, productivity, and human flourishing? Whatever the answers, one thing is certain. Aging is no longer an impenetrable wall. It is a frontier. And like all frontiers, it invites exploration, debate, and imagination. Senolytics, cellular reprogramming, and microbiome engineering are the tools with which we may redraw the map of human life. The quest to extend health span, to live not just longer but better, is no longer a dream whispered by visionaries. It is a scientific pursuit grounded in data, funded by billionaires, tested in labs, and soon perhaps prescribed in clinics. Whether we live to see 100, 120, or even 150 in the future, the direction is clear. The science of anti-aging is no longer about resisting death. It's about embracing vitality. And in that pursuit, we are only at the beginning.